Good morning, everyone. I'm so excited to be here with all of you. I hope you are very excited to be here today to hear from our amazing panel. As you know, we're here to talk to some folks here who have had some ARPA-H funding and who can talk about their experience. We actually have, it's sort of set up like two teams. So we're starting with our team from Instapath, and that's with Quincy Brown, who is the Associate Professor at Tulane and Chief Research Officer and co-founder of Instapath, and Mei Wang, CEO and co-founder. On the other side, we have our second team, which is, we have Paulo Garcia, who is CEO and President of BLASTID, and I'm so sorry. Eleftherios, Mylonakis, all right, Chair of the Department of Medicine at Houston Methodist. And my name is Tina Reed. I am Senior Healthcare Reporter at Axios and author of the Vitals newsletter. So we're going to start off a little bit talking about each one of your companies. We're going to start with you. And I wanted to hear a little bit about your origin story and how you came to be this company that has gotten this huge grant from ARPA-H. So let's talk a little bit about how this started in the lab. What is Instapath? What are you doing? And where did it come from?
Sure. Yeah, so I want to thank you all for being here, and thank the Portal and others for inviting us. And yeah, we're really excited to be part of this, I mean, really historic initiative by President Biden in terms of the cancer moonshot. And ARPA-H is really sort of transformational sort of funding model. And so we're part of the Precision Surgical Interventions program through ARPA-H. PSI program, and so this is really about, they asked the question, what if surgeries were fixed flawlessly every time, and really in terms of cancer? And so I started working on this problem, you know, how do surgeons know when they've removed all of a tumor back in 2005 when I was doing my postdoc at Duke University. And I was using a technology then, a spectroscopy technology that had promise, but really we were trying to solve a pathology problem, which is looking at cells at the cellular level, morphologically, but doing it on the fresh tissue, but you know, trying to do that in the operating room. And so really, this problem has been sort of a white well for me. It's a really hard problem, actually. So I've been working on it ever since. So I came to Tulane in 2012, and Mei was one of my first graduate students. And so we had started working on a new technology of accomplishing this, doing very high-speed microscopy of fresh tissues near the patient in space and in time. And Mei and another student came to me and they said, hey, we want to start a company. And I said, that's great. Wait a year until, you know, I want you to be 6 months away from graduating because I want you to graduate for yourself, but also for me. And so they did. And so they did, in the meantime, they did NSF I-Corps, which is like a boot camp sort of for companies. And in the same time, we were raising NIH funding, and so we'd raised, you know, a couple million, $2 or $3 million in NIH funding to move the technology forward. And so really there was this confluence of factors that happened around 2017 when Mei started Instapath. And so maybe I'll just let you kind of describe how Instapath started, and then, you know, sort of skipping forward to today, you know, now we came together as a team to compete for this ARPA-H award, and so yeah.
Go ahead, Mei.
Yeah, so we started, so as he mentioned, really the starting point was the NSF I-Corps program. And that was, I felt like really transformed this from an idea in the lab to something that could be commercially available. And we did the whole process. Whoever starting a company haven't done it, highly recommend it.
I agree with that.
Yeah, and it's really helpful. It's like I felt like I started as a scientist, and then I finished with a very different perspective of how the whole world works almost. Yeah, and before it's kind of just like, oh yeah, you do science, you like make things work in the benchtop, and then it will somehow magically become available in the commercial space. And then you go through the I-Corps process, you're like, oh, it's not magic. It's really like there is a very set way you can kind of make it happen. And that's really, I feel like that's our origin story, is the I-Corps. And since then, we started the company. We had grants from NSF and CPRIT, and also we have participated in a few accelerators, and that all has been like propelled us to this point from like where we were. We are very thankful to be part of all those teams, and so many people have helped us to get to where we are today. But I feel like if you want me to pinpoint a point, that's where everything started.
How much funding have you raised to date?
I haven't counted the ARPA yet. Before ARPA, it was like around, I think it's $5 to $7 million.
And then there was the 23 million from ARPA-H.
That's mostly.
Yeah, so that came to Tulane. So I'm the PI of that ARPA-H at Tulane, and then we have University of Georgia, University of Utah, and Instapath. And so really this PSI model is, develop a technology and then translate it as fast as possible. And so I think it helped us. And if I look at some of the other folks who were funded in this space, a lot of them were university academic industrial partnerships, right? So it's people like us who had a research lab and a company that had spun out, who are really working toward the same goal. But the issue that we had was, the technology profile was not so. What Instapath does is, you can image a skin resection in a couple of minutes, right? And there's a clear sort of market value there. But when it comes to scanning a very large tumor resection, we were not fast enough to do that.
How fast was it?
45 minutes.
Okay, so the problem with that is if you're in surgery.
If you're in surgery, that's too long.
Okay.
Right? And then also we were 45 minutes just to get an image, but then a pathologist had to read it. So what ARPA challenged us with is they said, do that 6 times larger area, twice the resolution, do it in 10 minutes and no pathologist in the room, and deploy it in 2 rural hospitals at the end of 5 years.
Sure.
And so that was the challenge they gave us. And so we saw the call and we said, this is crazy. This is impossible. And then a week later we had a plan. And so I really appreciated that model, which was like, it taught me something about thinking big and not being too conservative. And we get trained this way. We go to NIH and I love NIH, but it's not geared toward this type of a jump, right? So.
And what is the status? Where are you at right now in the commercialization journey?
Yeah, so Instapath is a company. They have a product, 2 products on the market.
We have a research grade product, and then we have a class 1 medical device, NEO. That's the one we can use for rapid on-site evaluation of any type of biological tissue from any procedure, really.
So slide-free digital pathology, right? So for digital pathology on a fresh sample. So that's continuing. But then with ARPA, the idea is that we're developing the next generation of the technology, which is still in development, right? And so we will see where that goes.
Great. Well, let's shift over to the BLASTID team. Now, this is a technology that's a bit cancer adjacent. Obviously, we're talking a lot about cancer today. And this is a Harvard spin-out. So we'll start with you, Paulo. Can you tell us a little bit about how this company came to be?
Yeah, thanks for the introduction. Paulo Garcia, CEO and President of BLASTID. BLASTID is a Harvard Medical School spin-out, specifically from Professor Johan Paulsson's lab from systems biology. And it came about as the grant was awarded, but it builds on decades of research and pioneering developments that have been advanced at Harvard in terms of being able to trap individual cells in microfluidic devices and imaging them in a time-lapse type of resolution. So this gives the ability to understand mechanisms. It gives the ability to interrogate what happens to the cells in the presence of different probes or reagents. And really, through the ARPA-H grant, which is a grant that funds 30 groups, 25 academic medical centers and researchers, 5 hospitals, and 3 companies, through this grant, what we're doing is we are developing a better fundamental understanding of what happens to these cells in the particular journey. We are developing a way to do new discovery of therapeutics. And BLASTID specifically is the diagnostic arm of the project in terms of building an in vitro diagnostic system that will be installed at 2 hospitals, MGH and Houston Methodist, for eventually doing clinical trials with these technologies. So it is all about translational research, taking something that was developed at the university, continue to advance and develop it, and then to help patients in the long term. So that is the big picture of the grant.
From my end, I would like to take you a little further back. I think that finding the funding and creating a company is exciting, but is the end product of what happened before and how the concept and the idea and the excitement of people and their willingness to work together to solve a problem and help humanity comes up. And what is the issue sometimes is that you have clinical people and scientists working different pathways and in different silos. So for us, the idea and the concept started with discussions with Johan about how he was describing to me how he has developed this technology, and I was describing to him how I have this problem of diagnosis. People, we think that because there are antibiotics out there, because there is good treatment and supportive care and critical care, we believe that, okay, infections, we're going to give you an antibiotic and it will go away. Of course, a lot of people know about resistance, but some people don't realize that some infections, the mortality rate goes up by the hour. And anybody can have an infection. If you have a trauma, if you have a perforation, if you have an appendicitis, all of us will have an infection in our lifetime many times over. So the concept that I would like to bring to the attention of people is to have scientists and business people and clinical people come together. And that's where I think the excitement happens, because each one of those individuals has, or groups of people have something to contribute to make this exciting and make it more practical. I think that, I do believe that if you really solve a problem that will help humanity, people will fund you. And it's the zest and the excitement that you have that you are going to have something for other human beings that will help you, give you the drive to knock on doors and apply for grants and knock on doors and do it many times over until you have it funded and you have it outside of academia going into becoming a reality. So I think that the more communication, more we break our silos, more we try to have practical thoughts and practical, apply in a practical way those discoveries. I think that is where all big ideas start, or small ideas, or all the effort starts, and then the company.
And your idea started, or this idea started with using this platform as for antibiotic resistance, but then you think bigger to its application to cancer. I mean, can you talk a little bit about that?
Yes. What Johan was describing to me, it was very similar to what I was seeing in my patients, because usually you take human cells or bacteria and you grow them in a lab environment, in a very artificial environment. And he was describing how he can see every single cell in a flow, which is the way that cells and bacteria and everything goes through the human body. It's usually the capillary cells, the smaller cells, where single cells pass through. And he was describing how he can see that. And all his findings about those cells and how they changed by moving around that machine, it was very similar to what happens in human body. I was explaining what I was seeing with my patients. I was seeing the relapse rate. I was seeing bacteria that I thought they were sensitive and they're resistant, and I give antibiotics to my patients, and they don't respond. And why is this happening? And he was describing to me that, and the concept is not only, and that links to cancer, because obviously most of the patients who have cancer, unfortunately, they develop infections, and that's how it happens. And walking here this morning, I was thinking of a 30-something-year-old person that I was treating up in Boston 15 years ago, who had leukemia, and he was diagnosed with acute leukemia a year after he got married. And he had a fungal infection in his brain. And I still remember how empty I felt inside me. When I was walking in his room, I was looking at him, I was looking at his wife, and I knew that the fungus will kill him. No matter what I was going to do, it was going to be a slow, horrible death for this young person starting a new family. And this has stuck with me, and obviously with his family and with his wife and everybody else, for years and decades. And infections and cancer, they are linked together, not only because of the complication, but also because infection limit what you can do for the cancer treatment. So there is the link there, and obviously, if you isolate cells, bacteria or human cells, you can do whatever you want with them. You can look at them in the microscope, you can do assays, you can evaluate them, you can do diagnostics on them. So the other concept that I think is becoming more and more obvious in everybody who is working on this project is how we can spin this now to look into cancer cells also and predict cancer.
And then Paulo, I want to put a finer point on what you can do with your technology that couldn't be done before. We were talking about how much quicker you can work.
Yeah, so for us, it's all about speeding up the process of giving clinically actionable information to the physicians. It's all about being able to do something that currently takes days and shrink it down to minutes or a couple of hours, while there is still room to adjust treatment, to provide the right dose, to de-escalate maybe some of the broad spectrum therapeutics that are being used. And we're doing that because we're merging 3 different technologies together. We are certainly doing advanced microfluidics, so think about being able to take a whole blood sample, much larger volume than others have tried, so around 10 milliliter, processing that quickly and being able to separate red blood cells and white blood cells from the cells of interest. And those cells, in the initial case, will be pathogens, but it could be other cells, such as circulating tumor cells, to link it back to this application. So now you have the ability to isolate these bacteria or organisms in a single cell manner, but being able to do high throughput interrogation, sometimes in the hundreds of thousands or millions of cells. So you can do this via super resolution microscopy, and so there are ways to increase the resolution that you can get so that you can get sub-micron features on the cell. This helps in terms of identification of the type of cell that exists, or measuring any changes that the cell exhibits as they get exposed to the different panels of antibiotics that we're testing. And we're leveraging machine learning and AI to help us detect those changes and to help us train and be able to provide information to the physician that could be, is there a pathogen present? Yes or no? Is it a gram-positive or gram-negative? Yes, or which one it is? We can go down, and the goal is to go down to the species level, so that something that is currently taking days can be done within a couple of hours. So that's the type of speed up that we're trying to do, and we're making great strides in all fronts. Again, leveraging all of these experts in the world that are part of the ARPA-H grant that we have constant access to, that we have reporting to do together, that we have active collaborations with. And that's the beauty of what ARPA-H has enabled for us, is this environment that is breaking down existing silos. You know, we're reporting on data that sometimes was generated 2 days ago. And so we're doing this in a way in which we're all working towards the same goal. The same goal is to help the patients to really advance the fundamental understanding and translate these technologies for the betterment of humanity.
How much funding have you raised to date?
The whole grant is $104 million. That is spread across the different groups. BLASTID itself has access to around $20 million directly, and additional funding through a lot of the efforts that would benefit us, but that are going to other groups.
Great. So going back to the Instapath team, one of the things I find interesting about ARPA-H is that they do separate into all these different programs, and they put out their challenges in a really tangible way. So they'll say, we're having a project to cure blindness, or automatically fight cyber attacks for hospitals. And you were describing how your project, or the program that you're part of, is to perform surgery flawlessly the first time. Can you talk about how did you sort of get ARPA-H's attention? Did they have that question sort of out there and you applied? Can you just talk about that process of how you became part of that program?
Sure. So, so this was, I think, maybe the second program that ARPA-H had put out. So before the programs, they had what they was back then was an open BAA, and so you could just come with an idea and you could get funding for that. But then they started to develop these programs, and you kind of mentioned a few of them. And I think PSI was one of the second ones they came out with. And so the program manager was someone who actually knew through NIH. She worked at NIH before. And so I got an email from her as she was leaving NIH. I think she sent it to a bunch of people and said, hey, I'm going to ARPA-H, and be on the lookout for some announcements later this summer. And so that was in April. And then I think in August, the announcement came out. And the announcement was a prescription. It basically said, here's, there's 3 topical areas, technical areas, where 3 challenges that we want to solve. Here are the requirements. Here's what you have to be able to do. And we had, I think, 9 weeks to put the proposal together. We had, there was, you had to compete for an abstract phase, and then if you were selected, you had 9 weeks to draft the full proposal. And so, again, I'd been working in this area for a long time. Instapath, we'd already formed, and so we were coming together as a team as it was, but we didn't have all the expertise that we needed. And so, for instance, the AI ML part, they wanted, there were certain types of cancers they wanted us to look at, so I needed clinical expertise that I didn't have. So really it's a partnership between engineering, computer science, entrepreneurship, and product development, quality systems. They want all the documentation done for FDA, and the clinical side. And so it really allowed us to, I just went out and said, hey, I put this team together really quickly, people that I knew and had worked with, but in different areas. And so I think we just came in with a really good package in terms of the right expertise, kind of Paulo was talking about, again, building really good teams that have expertise across all the disciplines that are necessary. So. And
how do you bring this team together? How do you convince people to come on board? I mean, are you hiring them? Are they just sort of saying, okay, let's see if we can work together, and if we win, what does that look like? Yeah, it was kind of the latter, right? So it was just going to them and saying, hey, look, there's this great opportunity. I think, here's a vision for what I think we could do together. And thankfully, just everybody said yes. And so I think what I really love about this, one of the things I really love about it is this team is fantastic. Everybody's great to work with, and really is focused on the science and moving this forward to help patients. And so, yeah, they all just said yes, but they could have said no, and that would have been bad. But they all said yes, so very thankful. What do you think was the X factor in terms of winning ARPA-H's attention, getting the funding with the package that you put together? Either one of you, whichever one of you wants to take it. I mean, our case is a little different for the programs because they know what they're looking for.
right? So they have a goal, and they have milestones that are set out, and they say by year 1 you have to be able to do this, and by year 2. And so for us, the target was known. How we got there was the question. And so I think I think it was just, again, like I said, we initially thought this is impossible, and it may still be, but we, within a week or so, we had a really good plan. And so I think it's, think big. I think, I don't think if you came in and said, well, realistically, based on what I've been doing the past few years, I think this is what you could achieve. No, I think you need to try to achieve what they want. And they understand that not everything's going to hit the mark. But if we don't really take big swings at things, we're not achieving the mission of ARPA-H. And so I think my advice would be to take big swings, but back it up with good science and good teams. Yeah.
Maybe to piggyback on that, it needs to be a vision that is transformational. It cannot be incremental. It's a vision that's transformational, that is going to impact, tens of millions, hundreds of millions of people, because that's what ARPA-H is about. It's about being able to impact all Americans, and that's one of the things that Johan actually is phenomenal at in painting a very, very ambitious project, an idea, and then let's figure it out as we go. If A, B, and C work independently, we should be able to make them work together and create something that is way beyond, the additive. And so that transformational, I think, is one of the key things here. And ARPA-H was created to enable this type of transformational change. So that is the right bet to place.
And your entry into ARPA-H was a little different because you weren't already an existing company. You were actually created by ARPA-H. Is that correct?
As a company, BLASTID didn't exist before the ARPA-H grant was funded, but there was a relationship that existed from before, and these projects or the technology that we are licensing from Harvard actually has been funded before through DARPA, the BTO, also through NIH. So there is a lot of demonstrations and scientific advancements that we are, starting from. And BLASTID specifically, what we're doing is we're focusing on the diagnostic side of things. So taking as many of those relevant advances and integrating them into a system that will be deployed in the hospital. So we're building hardware, we're building software. We will have a consumable cartridge. We're looking at how to do buffer exchange. Quality management systems will be part of the journey. And those are things that you're not necessarily thinking about in academia, but that are a requirement to be able to build something that it will be safe for patients and that could have the potential for medical impact. So that is the journey.
Yeah, I think that ideally you would like these connections to pre-exist. And yes, having everything under one umbrella would have been ideal. But this is not easily achievable. Here you have 10,000 beds next door. You can walk and there are 10,000 beds, all the hospitals combined. There are 4 or 5 different universities and research institutes just in walking distance from here. But this is a unique environment. And this discoveries that will happen here will not necessarily apply to rural areas or to areas that have modest means. I think that events like the one that we're having today signify the need for better communication between different disciplines that need to work together. In order to get a big change in how science and medicine functions, you have to have talents from different groups of people. And because Big Pharma cannot really accept the level of risk that academic labs and ARPA-H can accept, then it's up to us and up to individuals to try and make those connections and try to put together those groups that can synergize and create those big concepts and make them a reality.
And can you talk a little bit more about like how does the collaboration with Houston Methodist add to the project's goal of getting to the patient?
Yeah, well, when we started working on that, I was still in New England, so it was the proximity, the geography helped. But when I came here, I found a place that clinical trials can happen very quickly. And there is a whole group of biostatisticians, research nurses, research personnel that you need that is supported by the institution. This is a unique environment that really helps get clinical trials done without the research team having to spend the amount of money that usually you have to spend for a clinical trial. There is a pre-existing organizational support for a clinical trial. So obviously that was a welcoming news to the rest of the group. So that really helped quite a bit. So you need the access to patients and to patient samples in a way that doesn't really take all the resources out of the science.
And Quincy, can you talk a little bit about how the work at Tulane, or how your work at Tulane relates to the work at Houston Methodist?
Well, we're not at Houston Methodist, but we do have clinical partners in New Orleans. So the New
Send
Orleans
us
VA.
your device and we'll test it here.
Actually, we could be. We should talk.
We have tried.
Yeah, they could be. Yeah, we should talk. So Instapath is actually headquartered here, and we have done quite a bit of work at TMC. But yeah, so we have clinical partners at the New Orleans VA. And also a general hospital, so a community hospital in New Orleans. And so I think one of the things that may have helped with our project, so Instapath had already been working on cost reduction for their systems and had been working in rural hospitals. And so had deployed systems to rural hospitals in West Virginia and Pennsylvania and other places, and actually in Texas. And so part of the vision of ARPA-H and Not the Cancer Moonshot from President Biden is for every American, right? And so that was made very clear in the beginning, was that you can't build something that can't be deployed in a rural hospital. And I grew up in a small town in North Louisiana, so for me that's very poignant, right? So that I agree with that philosophy. And so that was something we had already been doing, the company had already been doing. And so we went into this project with that as a mindset, saying, how are we going to deploy these systems in community and rural settings, and perhaps even global settings? And so we built that into our clinical partnerships up front.
And you have like a device that actually, it looks like a suitcase-sized device, doesn't it?
Yeah, right now it's about a suitcase. Well, there's a smaller one. Yeah, there's two. Yeah, there's one that's about the size of a suitcase, and one that's the size of.
It's smaller. It's a printer size, I would say.
Printer size?
Yes.
Like vertical printer?
It all depends on the size of the suitcase. It can be a small suitcase.
Yeah. But yeah, and then the new system that we're building remains to be seen.
Okay.
But yeah, the idea is it could be mobile, right? It could be wheeled into an operating room or adjacent to an operating room. And we're kind of solving similar problems in different spaces. We're trying to take this process of histopathology that normally takes days, and we could do it in 45 minutes, but that's still not fast enough, so we need to do it in 10. It's really, so we're using super resolution, similar things here, but really totally different goals. So I think, there's been a huge push for early diagnosis, right? So early diagnosis is important, but we lose the value of early diagnosis if we don't do good surgery. And so I really feel like this, they picked a great problem to work on. Progress had stalled, I would say. And so I'm hoping that this will break down those barriers. So.
And just to make sure we didn't skip over the piece that I think is really interesting, when you were working with your community partners, you're making sure a rural doctor, somebody doing a surgery in a rural hospital, not some big fancy city hospital, is able to use this technology. They've got the AI.
Yes, so they not only use it, but deploy it, right? And so the AI is a huge part of it, right? So we anticipate that it's possible we could capture 1 trillion pixels of data in 10 minutes.
Wow.
Okay, so 1 to 4 terabytes of data. And then we somehow need to classify that data and then deliver it to the surgeon to make an action in 10 minutes, but without a $2 million GPU cluster, because they're not going to have it, without cloud computing necessarily, because the internet's not great.
Yeah.
So we have a team at the University of Utah who are experts in cyber infrastructure, AI, ML, really like the hardware, software interface that's needed to try to figure out how to solve this problem from a computational perspective. And so that's a big part of the project that we have. So.
I think the cost effectiveness side of things and being able to provide access in rural areas and other geographies is one of the key things that ARPA-H is also helping to enable. So we're the same. You know, some of the commercially available solutions out there for imaging could be $1 million, $2 million type of systems. We're trying to build them for, you know, 50,000, 100,000. So bringing down the cost by a factor of 10 or 100 if we can. So that is certainly one push for those of you writing grants to ARPA-H. That is one of the emphasis. The other one is talking about the clinical trial workflow is to, as soon as possible, even in your preliminary data, to start generating data with real samples. And this is where the samples that Dr. Mylonakis' lab is collecting now and shipping to Harvard for testing, it's so valuable, because the last thing you want to do is to build a platform that uses pristine samples in, you know, perfectly characterized buffers, and then you get a blood sample and then everything breaks. You want to make sure that you understand how to handle that along the way in partnership with your collaborators. And that's where frequent checkpoints, access to data, brainstorming sessions, that makes the difference. And with ARPA-H, we're doing this on a monthly basis. We have monthly meetings in which we're reporting commercial progress, and we have some of the academic groups attend. We are attending the academic groups reports as well, and we have the 2 annual meetings. So there is a lot of opportunity to just learn, be aware of what is happening, and establish collaborations to continue advancing the goal of the project. And there's been some nice surprises, and those nice surprises actually enable you to then push even further. And for us, that has been in the sample prep side of things.
Yeah, just to add to that, like also for somebody developing solutions like ours, we're scientists, we're not physicians, so we're technically not the users of these technologies, and we're also don't completely understand the healthcare system. I don't think anybody completely understands the healthcare system. Anybody?
Fair, that's fair.
Right? Like, I think talking to clinical partners is so important. You kind of get more information from a world you don't really know. And you don't step in their shoes every single day. You kind of have an idea. But it's kind of important to check in every day, like not every day, but like every week or so, like, are we still designing this for you? Because sometimes when you start designing things, you're like, oh, maybe we deviated from what we initially thinking about, because we forget some small details, and those details really only your clinical partners can provide those to you.
Yeah, and I'll just add, across a diversity of clinical sites, right? Because
Yes.
they're, academic medical center versus
Yes.
the VA versus
Yes.
a community center, it's very different, right?
Yeah, as he said, not every hospital have 10,000 beds. Those are very, very different from like the clinics and rural area. Just we learned those very quickly. Like, everybody are operating in very different resources, and it's not like one size fit all.
Well, we're coming up on time, but I did want to end on a question about looking forward to what's next. What should we be watching for in the world of cancer research, in the world of research, and in the funding environment, especially as we look at the fact that we're at a time of transition in leadership. We're seeing a whole new administration, obviously new leaders. We know who the nominee is for the HHS. So as you look ahead the next 5-10 years, I guess, what is the biggest thing you think people should be watching for in terms of cancer research funding and just the movement of cancer research?
Yeah, so I mean, so cancer is not going anywhere. I mean, I hope it does, but it's that's the the job of the scientific and clinical community is to make progress. So I feel optimistic that funding for cancer will continue. I do think that the model of ARPA-H is one which really promotes a translational mindset, which I think is important, and I think my perception is that it would be viewed on favorably by the next administration. But I think that we have to be careful that we don't do things at the expense of basic science, and so I think basic science can get lost in the shuffle sometimes when certain types of oversight say, well, I don't really understand why they're doing that, and the reason is because we may not know why it's useful yet, but one day it will be transformation. We saw that with the vaccines, right? And so, so I'm trying to remain optimistic, but we'll see. Yeah.
Mei?
I think here the theme here is just faster, more instantaneous. That's really, I think, what I see is going. AI, of course, and those are really major topics, but just automation, AI, those are just the things I think next 5 to 10 years a lot of funding and research will be going into.
And Paulo?
Yeah, I think part of the beauty of these type of spaces and demonstrations is bringing together different disciplines that can help advance the impact for patients with innovative technologies, and what we will continue to see is how to make those even more accessible to patients. That's really what, through ARPA-H and other grants, private, public type of funding, will be enabled, really the translational side of things.
You're asking us to predict the future, and that is a little difficult. I think it's impossible to predict what exactly is going to happen, but it's reasonable to expect that funding will suffer. Funding for research is the first one that goes out the window when there is any cuts, especially if we're not going to touch structural parts of the budget. So it's reasonable that we'll need to be more innovative, more inclusive. I hope that philanthropy will come and bridge some of the gap. Houston is, since we're talking about our environment here, is #1 city in philanthropy year after year for the last decade or more. This is an example that I suggest that others consider also to support and bring ideas into reality. I think that I'm very bullish on the infrastructure that is built in this environment. Now I'm going to walk back to an honor roll hospital, and in order to get there, I'll pass the #1 hospital for cancer and the #2 hospital for children care and 3 universities, just to walk to the hospital. I'm excited also about this community of small startups that is getting built and leverages academia. If you ask me, the biggest concern that I have is that big pharma continues to be very risk averse, and that's the result, in my mind, more of a downside for our ability to help individuals and patients than what government is going to do. Hopefully we'll be able to come together as groups of academicians and scientists and entrepreneurs and engineers and solve problems and help our patients who unfortunately have to suffer. Thank you.
Thank you so much. And thank you so much to our panel. Thank you so much for being such a wonderful audience. That's all the time we have.